Biochemistry 401, Lecture 24. Today we're going to talk about the regulation of glycogen metabolism. We're going to discuss both local signals and global signals that affect glycogen metabolism. So let's get started. Now, if you'll recall, glycogen catabolism is catalyzed by the enzyme glycogen phosphorylase, and this is one of the primary sites of regulation for this pathway. The glycogen phosphorylase enzyme is shown here. It is a homodimer that's allosterically regulated. One of the subunits of this dimer is shown in yellow and the other one is shown in white. Now, this enzyme is generally found attached right to glycogen, but its activity is dictated by its phosphorylation state and also by the presence or the absence of non-covalent allosteric regulators. Now, glycogen phosphorylase enzyme exists in two different states, a tense state and a relaxed state. The tense state is less active and the relaxed state is more active. Now the thing is, the enzyme shifts back and forth between the tense state and the relaxed state. But different factors can shift the equilibrium between the tense and the relaxed state to favor either one or the other conformation. Now in the tense state, the active site for the enzyme is partially blocked by an alpha helix that extends into the active site. When the enzyme is phosphorylated, this helix is kept out of the way, and so the enzyme is more apt to be relaxed. When phosphorylated, glycogen phosphorylase is known as phosphorylase A, and in this phosphorylation state, the enzyme is more apt to be in the relaxed state, whereas when glycogen is not phosphorylated, is known as phosphorylase B. And in this state, the enzyme is more apt to be in the tense state. And we can see this in the figure. Now this figure is a little complex, but let me walk you through it. In the upper panel, we see phosphorylase B and phosphorylase A, both of which are in the relaxed state. And in the bottom of the diagram, we see glycogen phosphorylase B and A when they're in the tense state. Now if you look at the length of the arrows that lie between phosphorylase B in the relaxed state and in the tense state, you can see that the arrows indicate that phosphorylase B is most often in the tense state. And in this state, the alpha helices extend into the active site, and so phosphorylase B is more often tense and more often inactive. In phosphorylase A, on the other hand, the arrows are shown to favor the relaxed state. And so when glycogen phosphorylase is phosphorylated, the conformation exists such that it favors the relaxed state. And in the relaxed state, glycogen phosphorylase is more active. As we shall see in a minute, the phosphorylation of glycogen phosphorylase occurs in response to global signals. And so to recap, when glycogen phosphorylase B becomes phosphorylated, it is known as glycogen phosphorylase A. Glycogen phosphorylase A is more apt to be in the relaxed conformation, in which the active site is open and accessible. When glycogen phosphorylase A becomes dephosphorylated, it is known as phosphorylase B. And again, in this conformation, glycogen phosphorylase is more apt to be in the tense state, in which an alpha helix extends partially into the active site, and so glycogen phosphorylase will be less active. Now the phosphorylation of glycogen phosphorylase happens by way of glycogen phosphorylase kinase. So far we know that glycogen phosphorylase is regulated by phosphorylation, such that when phosphorylated, phosphorylase B is turned into phosphorylase A, which is the more active conformation. But what phosphorylates this enzyme? Well, that is phosphorylase kinase, that's shown here. This is a homotetramer consisting of alpha, beta, gamma, and delta subunits, 4 of each. and it's arranged in a molecule that looks like a butterfly. Now, in this homotetramer, the subunits have different functions. Gamma is catalytic. The alpha, beta, and delta subunits are regulatory. The beta subunit is a phosphorylation site, and the delta subunit is a calmodulin homolog. It binds calcium. Both phosphorylation and calcium binding partially activate the kinase. However, when calcium is bound and when the beta subunit is phosphorylated, phosphorylase kinase is maximally active. And so now we have two pieces of the puzzle. We know that glycogen phosphorylase B is activated when it's phosphorylated to become phosphorylase A. And we also know that this happens through phosphorylation through the action of phosphorylase kinase. We also know that phosphorylase kinase is activated by both phosphorylation and by calcium. So now what we need to know is what phosphorylates phosphorylase kinase, and also how is it that there's an increase in calcium in the cell. This happens by two different methods, through global regulation and local regulation. In the global regulation in muscles, this phosphorylation occurs through the action of protein kinase A, pKa, and the local regulation occurs through an increase in intracellular calcium, and this happens in response to muscle contraction. When muscles contract, one of the things that happens is calcium is released from intracellular stores to allow this contraction to actually occur, and so muscles will have an increase in intracellular calcium when they're contracting. And let's face it, if your muscles are contracting, that means you're going to be burning energy, and you're going to need glucose. And so this is a really good signal. In the liver, the global regulation is by phosphorylation through protein kinase A also, and also through an increase in intracellular calcium. So now we have another piece of the puzzle. But how is it that pKa becomes activated? What about those global regulators? Global regulation occurs through two different signals, epinephrine, which is also known as adrenaline, and glucagon. Now, epinephrine is released from the adrenal glands that are shown here in yellow. These glands sit on top of the kidneys and are about the size of a walnut. Epinephrine is released from cells that are found in the adrenal medulla. This is the central portion of the adrenal glands, in response to a fight or flight mechanism that's initiated. And so when do you release epinephrine? Well, you release epinephrine during the fight or flight response. This can be activated when you're really hungry or when you're really upset and afraid of something. So I don't know if you've experienced this, but sometimes when you have gone without food for a while, you can become agitated. You can see this in children quite often because they become especially cranky when they're hungry. And this actually is the body's response to hunger. Your system is trying to get you moving so that you go and find food. And also when your glucose levels are low, glucagon will be released from the alpha islet cells in the pancreas. And then glucagon can bind to its receptors and activate mobilization of glycogen stores. So how is it that glucagon in the liver and epinephrine in the muscle in the liver have an effect on glycogen breakdown? When these global signals bind to their receptors, they cause a conformational change to occur in the receptor such that the alpha subunit is released from the beta and gamma subunits, and the alpha subunit then binds to adenylate cyclase and activates it. Once activated, adenylate cyclase synthesizes cyclic AMP from ATP. An increase in the concentration of cyclic AMP in the cell results in the activation of protein kinase A, and protein kinase A phosphorylates phosphorylase kinase and activates that. phosphorylase kinase then phosphorylates phosphorylase B to form phosphorylase A. This is the more active form of the enzyme. And phosphorylase A breaks down glycogen to release glucose 1-phosphate. The muscles themselves have local control of the release of glucose from glycogen. When you're exercising, your muscles actually release calcium from intercellular stores as part of the contraction mechanism, and this results in increased levels of calcium in the cell. Increased levels of calcium can activate phosphorylase kinase, which can then phosphorylate glycogen phosphorylase to activate it and thereby release glucose 1-phosphate from glycogen. And also, when one is exercising, or when one even anticipates exercising, epinephrine is also released. Now, the thing is, you sure don't want to be breaking down glycogen and making glycogen at the same time. And so, glycogen phosphorylase and glycogen synthase are reciprocally regulated. Now, in this slide, those things that are shaded in blue are active. Those things that are shaded in red are inactive. Now, protein kinase A phosphorylates many targets within the cell. One of them is phosphorylase kinase, and another is glycogen synthase. We saw that when glycogen phosphorylase kinase is phosphorylated, it's active. Protein kinase A can also phosphorylate glycogen synthase and inactivate that enzyme so that we do not make glycogen at the same time that we are breaking it down. And this is an example of reciprocal regulation. The same signal activating one pathway and inactivating another. Now, glycogen phosphorylase in the muscle can also be allosterically regulated. High levels of AMP in the muscle means that there are low energy stores in the muscle, and it's time to mobilize glycogen stores to be able to support muscle contraction. And so high levels of AMP will cause AMP to bind in the nucleotide binding site of glycogen phosphorylase. This causes phosphorylase B to favor the relaxed state, even though it's not phosphorylated. And so even when glycogen phosphorylase is dephosphorylated and therefore in the B state, this control can be superseded by low energy stores within the cell. AMP can bind in the nucleotide binding site and thereby cause glycogen phosphorylase B to favor the R state. However, when ATP is high, or when glucose 6-phosphate is high, this means that energy levels or the availability of glucose is high in the cell. And this means that added glucose mobilization is not necessary. And so ATP and glucose 6-phosphate can also bind to allosteric sites within phosphorylase B to further deactivate it and favor the T state. Now, in the liver, we have a particularly interesting scenario by which glucose can bind a regulatory site that causes glycogen phosphorylase A to favor the 10 state. Additionally, binding of glucose in this regulatory site moves the serine residue into such a position that it becomes an excellent target for phosphatase, which results in the removal of the phosphate. And so glycogen phosphorylase A in the liver responds to the high level of glucose by favoring the 10 state and also dephosphorylation of the serine residue. In fact, glycogen phosphorylase A is the glucose monitor within the cell itself. This enzyme is highly responsive to local glucose concentrations, such that high levels of glucose in the cell will shut down this enzyme and prevent it from releasing more glucose 1-phosphate from glycogen. And so to recap, when glucose levels are low, glucagon will be released from pancreatic alpha cells, When one is exercising, or when one even anticipates exercising for that matter, epinephrine can be released from the adrenal medulla into the bloodstream, and this in turn can cause activation of glycogen phosphorylase and mobilization of glucose stores. It is important that you know this slide. When glycogen phosphorylase is inactivated, we have activation of glycogen synthase. This occurs through the action of protein phosphatase 1 that inactivates glycogen phosphorylase and phosphorylase kinase as well. When glycogen synthesis is required, protein phosphatase 1 is activated. This phosphatase removes phosphate groups from glycogen phosphorylase kinase and also from glycogen phosphorylase A to yield glycogen phosphorylase B. Protein phosphatase 1 can also remove phosphates from glycogen synthase, and in this case, it has an activating effect. And so glycogen synthesis will be stimulated and glycogen breakdown will be inhibited. Activation of glycogen synthase happens after a meal or when you're resting.
Insulin is released from the pancreatic beta islet cells in response to high glucose levels in the blood. Insulin will bind to its receptor, causing dimerization of the receptor. This results in the receptor becoming phosphorylated. This phosphorylation allows adapter proteins to bind to the receptor, and the downstream effect is actually quite complex. But one of the things that results in is the ultimate activation of protein phosphatase 1. And when protein phosphatase 1 is activated, it can dephosphorylate glycogen synthase to activate that, and glycogen phosphorylase kinase to inactivate that. And also, glycogen phosphorylase A will become dephosphorylated to form glycogen phosphorylase B, and thus glycogen phosphorylase is also inactivated. So the ultimate result of insulin binding is going to be an activation of glycogen synthesis and an inactivation of glycogen breakdown. This makes sense. After a big meal, you want to take that extra glucose and store it for later, and so glycogen synthesis is going to be active. This marks the end of Lecture 24. Have a great day.